Products Advisory Committee meeting. I'm Mike Kaczynski, Project Manager with FDA, and I will be today's meeting facilitator. This is a live public meeting that is being broadcast in its entirety through C-SPAN, Yourcast, Facebook Live, YouTube, and Twitter. Today's event is also being recorded and will be posted on FDA's VRBPAC webpage, along with all relevant meeting materials. Throughout today's meeting, I will be reminding our presenters, committee members, sponsors, and OPH speakers as to when they are close to their allotted time and assisting them when needed. Just a reminder to everyone that once called upon to please manage your mute and activate your webcams. At this time, I'd like to introduce you to Dr. Arnold Monto, the acting chair, who will now provide opening remarks. Dr. Monto, take it away.
I'd like to add my welcome to this 162nd meeting of the Vaccines and Related Biologicals Advisory Committee of the FDA. We have one task ahead of us today, and that is to discuss and vote on the emergency use authorization of the Pfizer BioNTech COVID-19 vaccine for the prevention of COVID-19 in individuals 16 years of age and older. To kick the meeting off, I'd like to call on Dr. Atreya to go through the roll call, introduction of committee, and administrative statements. As we go round the committee, I'd like the members to just introduce themselves and their affiliations. So Dr. Atreya, please.
Good morning, everyone. This is Dr. Prabha Atreya, and it is my great pleasure to serve as the designated federal officer as our DFO for today's 162nd Vaccines and Related Biological Products Advisory Committee meeting. On behalf of the FDA, the Center for Biologics Evaluation and Research, and the VRBPAC Committee, I would like to welcome everyone for today's virtual meeting. Dr. Monto already mentioned the topic for today's meeting is Emergency Use Authorization of Pfizer BioNTech COVID-19 vaccine for the prevention of COVID-19 in individuals 16 years of age and older. Today's meeting and the topic was announced in the Federal Register notice that was published on November 27, 2020. Now I would like to introduce my excellent staff and also to make a few administrative remarks. Ms. Kathleen Hayes is my co-designated federal officer providing support today in all aspects of conducting this meeting. Other staff, Ms. Monique Hill, Dr. Janet Devine, and Ms. Christine Wirt also provided excellent administrative support. Please direct any press or media-related questions for today's meeting to FDA's Office of the Media Affairs at fdaoms@fda.hhs.gov. The transcriptionist for today's meeting is Ms. Allison Bean. We will begin today's meeting by taking a formal roll call for committee members and temporary voting members. When it is your turn, please turn your video camera, then state your first name and last name and your organization. And when finished, you can turn your camera off so we can proceed to the next person. Please see the member roster slide in which we will begin with the chair. Dr. Monto, can we start with you, please? Thank you.
Right, I'm Arnold Monto. I'm professor of epidemiology in the University of Michigan School of Public Health.
Great. Dr. Amanda Cohn? Dr. Cohn, can you unmute your phone? We couldn't hear you, Dr. Cohn.
She'll be on shortly. She's reconnected. So we'll go to the next one.
Dr. Chatterjee.
Good morning. My name is Archana Chatterjee. I'm the Dean of the Chicago Medical School and Vice President for Medical Affairs at Rosalind Franklin University of Medicine and Science. I'm a pediatric infectious diseases specialist by background and have an interest in.
Thank you. Dr. Cody Meissner. Dr. Meissner?
Thank you. I'm Cody Meissner. I'm a professor of pediatrics in the infectious disease division at Tufts University School of Medicine and Tufts Children's Hospital in Boston.
Great. Dr. Gans.
Good morning. This is Dr. Haley Gams. I'm a professor of pediatrics at the Pediatric Infectious Diseases at Stanford University. It's great to be here.
Thank you. Dr. Kurila, Mike Kurila.
Good morning. Michael Kurila. I'm the director of the Division of Clinical Innovation at the National Center for Advancing Translational Science within the National Institute of Health. I'm a pathologist by training, and most of my professional career has been involved in infectious disease, drug and vaccine development.
Great. Dr. Paul Offit.
Hi, I'm Paul Offit. I am a professor of pediatrics in the Division of Infectious Diseases at the Children's Hospital of Philadelphia and the Perelman School of Medicine at the University of Pennsylvania.
Dr. Annunziato, Paula Annunziato.
Good morning. I'm Paula Annunziato. I lead clinical global development for vaccines at Merck, and I'm here today as the non-voting industry representative.
Excellent. Dr. Mr. Sheldon Tobman. Mr. Tobman. Okay, Dr. Steve Pergam.
Good morning. Can you hear me?
Yes, we can.
Thank you.
Yes, thank you.
Yeah, good morning. My name is Sheldon Taubman. I'm an attorney. I represent low-income clients mostly in the health area. I'm employed by New Haven Legal Assistance Association, although I'm here today in my personal capacity as a consumer representative.
Great. Dr. Steve Pergan.
Hi, everyone. I'm Steve Pergam. I'm an associate professor at the University of Washington and Fred Hutchinson Cancer Research Center, and I focus on infectious diseases.
Mike, can we go to the next slide, please? Great. Dr. Fuller, Ovita Fuller.
I'm Ovita Fuller. Good morning. I'm Ovita Fuller. I'm an associate professor of microbiology and immunology at the University of Michigan Medical School and a member of the African Studies Center in the International Institute, and I'm a virologist by training.
Great. Dr. Kim, Captain Kim.
Good morning, David Kim. I'm the Director of the Division of Vaccines at the Office of Infectious Disease and HIV/AIDS Policy under the Office of the Assistant Secretary for Health.
Great. Dr. Eric Rubin.
Hi, I'm Eric Rubin. I'm Editor-in-Chief of the New England Journal of Medicine, a professor at the Harvard THGN School of Public Health, and an infectious disease clinician at the Brigham and Women's Hospital.
Excellent. Dr. James Hildreth.
Good morning. I'm James Hildreth. I'm the President and CEO of Meharry Medical College, and Professor of Internal Medicine. I'm a viral immunologist by training. Thanks.
Great. Dr. Janet Lee.
Good morning. I'm Jeanette Lee. I'm a professor of biostatistics at the University of Arkansas for Medical Sciences.
Great. Next slide, please. Dr. Juan Banaclo.
Good morning. I'm Juan Gea Banacloche. I'm an infectious diseases clinician at the Mayo Clinic in Phoenix, Arizona.
Great. Dr. Mark Sawyer.
Good morning. I'm Mark Sawyer. I'm a professor of pediatrics at the University of California, San Diego, and radiology in San Diego, and I'm a pediatric infectious disease specialist.
Excellent. Dr. Melinda Watson.
Good morning. I'm Melinda Horton. I'm an adult infectious disease physician by training, and I'm director of the Immunization Services Division at the Centers for Disease Control and Prevention.
Great. Thank you. Dr. Ofer Levy.
Good morning. My name is Ofer Levy. I'm the director of the Precision Vaccines Program at Boston Children's Hospital and professor of pediatrics at Harvard Medical School.
Great. Dr. Pamela McInnes.
I'm Pamela McInnes, retired Deputy Director of the National Center for Advancing Translational Sciences at the National Institutes of Health. Good morning.
Great. Dr. Patrick Moore.
Good morning. I'm Patrick Moore. I'm at the University of Pittsburgh Cancer Institute and the Department of Microbiology and Molecular Genetics.
Great. Dr. Ralph Tripp.
Good morning. I'm Ralph Tripp from the University of Georgia. I'm the chair of vaccine therapeutics there.
Okay. Dr. Stanley Perlman. We can't hear you, Dr. Perlman. Mike, can you adjust the volume?
Yeah, good morning. I am Dr. Stanley Perlman from the University of Iowa in the Department of Microbiology and Immunology and in Pediatric Infectious Diseases, and I'm a long-term coronavirologist.
Great. I think Dr. Amanda Cohn is available. Can she introduce herself just for a second? We can't hear you.
Can you hear me now?
Yes.
Good morning. Sorry for the technical difficulties. I'm Captain Amanda Cohn, Chief Medical Officer of the National Center for Immunization and Respiratory Diseases, and a pediatrician by training with expertise in vaccines and pediatrics.
Thank you so much. Okay, let's now introduce introductions of the FDA staff. First, I would like to introduce Dr. Peter Marks, the head of the Center for Biologics. Dr. Marks.
Hi, so thanks very much. Peter Marks, Director of Center for Biologics Evaluation and Research, and I just want to take a moment to thank all of the committee members as well as everybody who's tuning in right now who might not usually be viewing our usually sedate vaccines and related biologics products advisory committee meeting. Thanks very much.
Excellent. Let's move on to Dr. Marianne Gruber, Director of the Office of the Vaccines at CBER.
Yeah, good morning. My name is Marion Gruber, and I'm the Director of the Office of Vaccines Research and Review in the Center for Biologics and Research at FDA. And on behalf of my colleagues in the Office of Vaccines, I also would like to welcome the committee members, Pfizer BioNTech, as well as the public to today's meeting. I would like to take the opportunity to thank the members of this committee for taking time out of their busy schedule to provide their perspectives, their recommendations, and advice regarding the adequacy of the scientific evidence that will be presented by Pfizer and the FDA today to support a determination whether the benefits of Pfizer BioNTech's COVID-19 vaccine outweigh its risks to support authorization of this product under an EUA. FDA very much appreciates the committee's input on this very important topic, and I look forward to today's discussion. Thank you.
Great. Thank you, Dr. Gruber. Now I would like to acknowledge the presence of Dr. Celia Witten, Deputy Director of CBOR, and also Dr. Philip Krause, Deputy Director of Office of Vaccines, who may join later making remarks. So I will now proceed with the conflict of interest statement. Okay, so the Food and Drug Administration is convening virtually today, December 10th, 2020, the 162nd meeting of the Vaccines and Related Biological Products Advisory Committee under the authority of the Federal Advisory Committee Act of 1972. Dr. Arnold Monto is serving as the acting voting chair for this meeting. Today on December 10th, 2020, the committee will meet in open session to discuss the Emergency Use Authorization, EUA, of the Pfizer BioNTech COVID-19 vaccine for the prevention of COVID-19 in individuals 16 years of age and older. The topic is determined to be a particular matter involving specific parties. With the exception of industry representative member, all standing and temporary voting members of the WEDPAC are appointed special government employees or regular government employees from other agencies and are subjected to federal conflicts of interest laws and regulations. The following information on the status of this committee's compliance with federal ethics and conflict of interest laws, including but not limited to 18 United States Code Section 208, is being provided to participants in today's meeting and to the public. Related to the discussions at this meeting, all members, regular government employees and special government employee consultants of this committee have been thoroughly screened for potential financial conflicts of interest of their own as well as those imputed to them, including those of their spouse or minor children, and for the purpose of 18 U.S. Code 208, their employers. These interests may include investments, consulting, expert witness testimony, contracts and grants, cooperative research and development agreements or CRADAs, teaching, speaking, writing, patents and royalties, and primary employment. These may include interests that are current or under negotiation. FDA has determined that all members of this advisory committee are in compliance with federal ethics and conflict of interest laws. Under 18 U.S.C. Section 208, Congress has authorized the FDA to grant waivers to special government employees and regular government employees who have financial conflicts of interest when it is determined that the agency's need for a special government employee services outweighs the potential for a conflict of interest created by the financial interest involved, or when the interest of the regular government employee is not so substantial as to be deemed likely to affect the integrity of the services with which the government may expect from the employee. We have the following consultants serving as temporary voting members today. They are Dr. Ovita Fuller, Dr. Awan Benaklock, Dr. James Hildreth, Captain David Kim, Janet Lee, Dr. Ofer Levy, Dr. Pamela McInnes, Patrick Moore, Dr. Stanley Perlman, Eric Rubin, Mark Sawyer, and Ralph Tripp, and Melinda Wharton. Based on today's agenda and all financial interests reported by committee members and consultants, there has been only one conflict of interest waiver issued under 18 U.S. Code 208 in connection with this meeting. Among these consultants, Dr. James Hildreth, a special government employee, has been issued a waiver for his participation in today's meeting. The waiver was already posted on FDA website for public disclosure. Dr. Paula Annunziato is currently serving as the industry representative to this committee. Dr. Annunziato is employed by Merck. Industry representatives are not appointed as special government employees and serve as non-voting members of the committee. Industry representative on this committee is not screened for their financial interests. Industry representatives act on behalf of all regulated industry and bring general industry perspective to the committee. Mr. Sheldon Taubman is serving as the consumer representative for this committee. Consumer representatives are appointed special government employees and are screened and cleared prior to their participation in the meeting. They are voting members of the committee. Today's meeting has multiple external speakers. We have 3 speakers from the Centers for Disease Control and Prevention. These are Dr. Nancy Masonia, Dr. Aaron Hall, Dr. Anita Patel. Regular government employees have all been screened for conflicts of interest and have been cleared to participate as speakers for today's meeting. The guest speaker for this meeting is Dr. Stephen Goodman, a professor of medicine and Associate Dean for Clinical and Translational Research at Stanford University. He has been asked to disclose any financial interest he may have related to the product before the meeting. Disclosure of conflicts of interest for guest speakers follows all applicable federal laws, regulations, and FDA guidance. FDA encourages all meeting participants, including open public hearing speakers, to advise the committee of any financial relationships that they may have with any affected firm, its products, and if known, its direct competitors. We would like to remind standing and temporary members today that if the discussions involve any other products or firms not already on the agenda for which an FDA participant has a personal or imputed financial interest, the participants need to inform the DFO and exclude themselves from such involvement, and their exclusion will be noted for the record. This concludes my reading of the conflicts of interest statement for the public record. At this time, I would like to hand over the meeting to our chair, Dr. Manto. Thank you very much. Dr. Manto?
Thank you, Prabha. Here I am. Technical issues, which are going to be one of our biggest problems, I predict, during the day. Thank you for getting the meeting kicked off. I'd like first, and we're actually running a little early, to call on Dr. Doron Singh of the FDA to talk about the situation that we are facing and the presentation of a description of the emergency use authorization. Dr. Fink.
Good morning. I'm Doron Fink. I'm the Deputy Director for Clinical Review in the Division of Vaccines and Related Products Applications within the Office of Vaccines Research and Review, Center for Biologics Evaluation and Research at FDA. The VRBPAC last convened on October 22nd of this year to discuss the development, licensure, and emergency use authorization of COVID-19 preventive vaccines. Since that meeting, COVID cases and associated hospitalizations and deaths have increased substantially in the U.S. and worldwide. On November 20th, Pfizer submitted an emergency use authorization request for the Pfizer-BioNTech COVID-19 vaccine, otherwise known as BNT162b2. This is an mRNA and lipid nanoparticle vaccine administered as a 2-dose regimen 21 days apart. We'll be hearing more about the vaccine and its proposed use in later presentations. The use being requested for emergency authorization is for active immunization to prevent COVID-19 caused by SARS-CoV-2 in individuals 16 years of age and older. The information submitted with the request includes safety and efficacy data from a large, randomized, blinded, placebo-controlled phase 3 trial. And these data will be discussed in detail in our afternoon sessions. Today, we will be considering whether to make available to millions of Americans an as-yet investigational vaccine that has been developed, tested, and reviewed in record time, with additional testing still underway in ongoing studies. The American public demands and deserves a rigorous, comprehensive, and independent review of the data. And that's what FDA physicians and scientists, all of us career public health servants, have been doing over days, nights, weekends, and yes, over the Thanksgiving holiday. This is in addition to months of review work already completed on information previously submitted in preparation for an EUA request. FDA has been conducting its comprehensive review of the Pfizer-BioNTech COVID-19 vaccine EUA submission since its submission on November 20th. Our review has included verification of clinical data integrity of Pfizer analyses and also our own independent analyses using data sets that were provided in the submission. We have continued an ongoing review of chemistry, manufacturing, and control information, non-clinical data, and review of clinical assays, including information that was submitted shortly prior to the EUA request. We have been reviewing and revising, along with Pfizer, prescribing information and fact sheets for vaccine recipients and healthcare providers. These will be necessary to inform and instruct vaccine recipients and healthcare providers during use of the vaccine under EUA, and these materials are necessarily informed by our review of the data. We have sent and received back answers to multiple information requests addressed to Pfizer to clarify questions related to the data. And last but certainly not least, we have been preparing for today's VRBPAC meeting. Today's meeting, in which the committee will advise FDA with its own independent assessment of the data, continues FDA's commitment to an expedited review process that is transparent, scientifically sound, and data-driven. The legal authority for emergency use authorization was established in Section 564 of the Federal Food, Drug, and Cosmetic Act. This legal authority allows for FDA authorization of unapproved medical products or unapproved uses of approved medical products to address public health emergencies related to biological, chemical, radiological, or nuclear agents. Issuance of an emergency use authorization requires prior determination of a threat and declaration of circumstances justifying the need for an EUA to address that threat by the Secretary of Homeland Security, Defense, or Health and Human Services. To that end, Health and Human Services Secretary Azar issued a declaration on March 27th of this year justifying emergency use authorization of drugs and biological products to address the COVID-19 pandemic. Once that declaration has been issued, there are 4 criteria that must be met in order to issue an EUA. First of all, the agent referred to in the EUA declaration can cause a serious or life-threatening disease or condition. We know this to be true for SARS Coronavirus 2 and COVID-19. The second and third criteria are closely linked. There must be a reason to believe that the medical product may be effective to prevent, diagnose, or treat the serious or life-threatening condition caused by the agent, and the known and potential benefits of the product should outweigh the known and potential risks of the product. There are special considerations for a COVID-19 vaccine anticipated for widespread deployment to millions of individuals, and these will be discussed on my next slides. The final criterion is that there should be no adequate, approved, and available alternative to the product for diagnosing, preventing, or treating the disease or condition. At this time, the only FDA-approved product for COVID-19 is remdesivir. This is an antiviral agent that is approved for treatment of COVID-19, not prevention. Additional products have been issued under emergency use authorization but have not been FDA approved, and none of these products is authorized for use to prevent COVID-19. Thus, at this time, there is no adequate, approved, and available alternative to a COVID-19 vaccine for preventing COVID-19 caused by SARS Coronavirus 2. In October of this year, FDA released guidance outlining our expectations for submissions requesting emergency use authorization of COVID-19 vaccines. And these expectations were discussed at the VRBPAC meeting in October. There are 3 main areas covered by our expectations. First, data to demonstrate manufacturing quality and consistency. FDA has reviewed the manufacturing quality and consistency data for the Pfizer-BioNTech vaccine and found it adequate to support emergency use authorization of the vaccine. This will not be discussed in detail further at this meeting. Second, we expect clear and compelling safety and efficacy data to support a favorable benefit risk of the vaccine when rapidly deployed for administration to millions of individuals, including healthy people. And finally, we expect plans for further evaluation of the vaccine safety and effectiveness, including in ongoing clinical trials, active and passive safety monitoring during their use under EUA, as well as observational studies. In terms of clinical data expected to support an EUA submission for a COVID-19 vaccine, we expect a high bar for efficacy. Efficacy data from at least one well-designed phase 3 trial to demonstrate protection against SARS-CoV-2 infection or disease with a point estimate of at least 50% compared to a placebo. Additionally, the appropriately alpha-adjusted confidence interval lower bound around that point estimate should be greater than 30%. This is to ensure that a widely deployed COVID-19 vaccine will have an appreciable impact. In terms of safety data, we expect these data from throughout clinical development to evaluate reactogenicity, serious adverse events, and adverse events of special interest. And we expect that a high proportion of phase 3 study subjects will have been followed for at least 1 month after completion of the full vaccination regimen. We have an additional expectation for follow-up that I will explain on my next slide. We also expect to be able to review sufficient cases of severe COVID-19 that have occurred in clinical trial participants to assess for signals of enhanced disease, and also, if possible, to assess for preliminary evidence of protection against severe disease. We recognize that a planned case-driven interim efficacy analysis and associated safety analyses at the same time could provide data to support an emergency use authorization. We have explained that we expect these analyses to include a median follow-up duration of at least 2 months after completion of the full vaccination regimen. The reasons for this expectation are that, first of all, it allows time for potential immune-mediated adverse events to be evaluated, understanding that uncommon but clinically significant immune-mediated adverse events to preventive vaccines generally have onset within the first 6 weeks following vaccination. A median follow-up of 2 months also ensures that vaccine efficacy is assessed during the time when adaptive and/or memory immune responses, rather than innate responses, are mediating protection. And finally, this follow-up period allows for early assessment of waning protection and for assessment of signals of enhanced disease. Following issuance of an EUA for a COVID-19 vaccine, we understand and expect that further vaccine evaluation would be needed for ongoing benefit risk assessment to support continuation of the EUA. But equally important, further vaccine evaluation would be needed to accrue additional data to support licensure of the vaccine as soon as possible and/or to inform labeling. This further vaccine evaluation following issuance of an EUA would include longer-term follow-up for safety, including in larger numbers of vaccine recipients and in populations with lower representation than in clinical trials. Further evaluation would also allow for more precise estimation of vaccine effectiveness in specific populations and more robust assessment of effectiveness against specific aspects of SARS coronavirus 2 infection or disease, for example, asymptomatic infection. This further evaluation would also characterize the duration of protection, could investigate immune biomarkers that might predict protection, and of course would be ongoing monitoring for signals of enhanced disease. Issuance of an EUA for a COVID-19 vaccine would be contingent upon the ability to conduct further vaccine evaluation, which would occur through a combination of active follow-up of vaccine recipients under the EUA, passive monitoring for clinically significant adverse reactions using established reporting mechanisms, for example, the Vaccine Adverse Events Reporting System, observational studies, including those that leverage healthcare claims databases, and finally, continuation of blinded, placebo-controlled follow-up in ongoing clinical trials for as long as is feasible, and strategies to handle loss of follow-up in those trials. We acknowledge that placebo-controlled blinded follow-up cannot continue indefinitely as more information about a vaccine's safety and effectiveness becomes available. However, FDA does not consider issuance of an EUA for a COVID-19 vaccine to necessitate immediate unblinding of ongoing clinical trials or offering vaccine to all placebo recipients. Of course, trial participants may choose to withdraw from follow-up for any reason, including to receive vaccine made available under EUA. And it may be possible to offer vaccine to placebo recipients in clinical trials in a reasonable time frame that doesn't compromise the integrity of the clinical trial. And these considerations will be discussed further this morning. When an EUA is issued for a COVID-19 vaccine, it will specify conditions of use for which benefit risk has been determined to be favorable based on review of the totality of available data, including those populations to be included or excluded from the EUA, conditions for vaccine distribution and administration, and requirements for safety monitoring and reporting of adverse events. Vaccine made available under an EUA will also include provision of information to vaccine recipients and healthcare providers via prescribing information and fact sheets. These materials will describe that the product remains investigational, will inform about the known and potential benefits and risks, and will also make clear what are the available alternatives and the option to refuse vaccination. Once issued, an EUA may be revised or revoked for a number of reasons. First of all, if circumstances justifying the EUA no longer exist. Second, if criteria for issuance are no longer met. And third, for any other circumstances that arise that warrant changes necessary to protect public health or safety. These other circumstances may be based on new information concerning vaccine safety or effectiveness, vaccine manufacturing or quality, or COVID-19 epidemiology or pathogenesis. The agenda for today's VRBPAC meeting will include, following the conclusion of my talk, first, 3 presentations from the CDC, providing an update on COVID-19 epidemiology, plans for vaccine safety and effectiveness monitoring under an EUA, and operational distribution plans for the vaccine under an EUA. We will then hear about considerations for placebo-controlled trial design if an unlicensed vaccine becomes available. Following lunch, we will have an open public hearing, and then we'll dive into a discussion of the data. First, with a presentation by Pfizer, and then an FDA presentation. At the end of the day, we will have a committee discussion and a vote. We have 2 questions that we would like the committee to consider for discussion. These will not be voting questions. First, Pfizer has proposed a plan for continuation of blinded, placebo-controlled follow-up in ongoing trials if the vaccine were made available under EUA. We would like the committee to discuss Pfizer's plan, including how loss of blinded, placebo-controlled follow-up in ongoing trials should be addressed. Second, we would like the committee to discuss any gaps in plans described today and in the briefing documents for further evaluation of vaccine safety and effectiveness in populations who received the Pfizer-BioNTech vaccine under an EUA. Following discussion of these items, we will have a single question for the committee to vote on. The question is, based on the totality of scientific evidence available, do the benefits of the Pfizer-BioNTech COVID-19 vaccine outweigh its risks for use in individuals 16 years of age and older? This concludes my presentation. Thank you very much.
Thank you, Dr. Fink, for a very clear presentation. We have a fair amount of time before the next scheduled presentation, which is very good because I think there are some questions of clarification that the committee may have about our guidelines for discussion and for our eventual vote. So committee members, please raise your hands if you would like to ask Dr. Fink some specific questions. We don't want to start our discussion of the points raised by Dr. Fink, but just clarification about the characteristics of an emergency use authorization and the other guidelines. So please raise your hand. I may be having some technical difficulties, so Mike, you may need to recognize the person. Go ahead.
No problem. All right, the first one we have is Dr. Sawyer. Would you go ahead and turn your camera on?
Thanks very much, Dr. Fink. You mentioned that issues related to manufacturing can be one of the reasons to revise or revoke an EUA. What is the monitoring process going forward from this point with regard to manufacturing process?
Thank you. So FDA continues to engage with the vaccine manufacturer concerning manufacturing quality and control issues to ensure that these remain adequate to support use of the vaccine under EUA. I'll invite any of my other FDA colleagues to comment further.
Yeah, this is Marion. Can you hear me, Mr. Chairman?
Yes, we can. We can hear you.
Okay, thank you very much. So no, I just wanted to second what Doran just said. It is true that the monitoring of the CMC, that is the chemistry, manufacturing, and control information, that is currently available and will, and we of course will get additional data from the manufacturing, from the different manufacturing sites. We will review this information and we will work together with the vaccine manufacturer to make sure that the product that is made and generated is of adequate consistency and quality. So this is work that's going to be ongoing over the next month.
Thanks very much.
Okay, Arnold, are you able to see the, yep, Arnold, are you
No,
able to see the
I
attendee
can't.
list?
That side of my screen I see, but no, there's no activity there.
Okay, that's all right. So, while
Will
you want to have you
why don't you manage the calling on?
We'll take care of that.
Right.
All right, Dr. Corella?
you. I don't know why my camera is not working, but it says it's on. Doran, just trying to understand the question we're being asked to vote on is what Pfizer has requested, but if I understood your talk correctly, the FDA can limit the actual can place limits on the target populations or the specific indications regarding the EUA. My question is, how do you view the investigational status of the product under an EUA?
Thank you for the question. So a product that is made available under an EUA is an unapproved product. It has not been FDA licensed.
So let me just, if I can just follow up with that. So if you limited the EUA to a specific set of populations, even those populations that have the EUA, placebo-controlled trials would still be possible?
Well, we will have a discussion on those considerations later this morning, but in FDA's view, issuance of an EUA for a COVID-19 vaccine should not preclude the conduct of placebo-controlled trials. In particular, in situations where that vaccine made available under an EUA is available only in limited quantity.
Okay, I think I can see now. Thank you for fixing it. Dr. Lee.
So thank you. One of the questions I have is a little bit more general. I recognize we're considering the Pfizer product for an EUA today. However, future EUA applications, if, for example, there is a vaccine that is approved under a BLA, vaccine number 1, let's just call it that, will that make it more challenging for future products to request an EUA?
Right. So I think what you're getting at is the fourth criterion for issuance of an EUA, which says that there must not be any adequate, approved, and available alternative therapy.
Yeah, correct.
So first of all, if a vaccine is made available under an EUA, it is not approved, and so therefore that would not preclude issuance of an EUA for another vaccine. If a vaccine is approved by FDA, that would also not necessarily preclude issuance of an EUA for another investigational COVID-19 vaccine. For example, if the approved vaccine is available only in limited quantity, then it may not be considered adequate to address the public health emergency. Second of all, if the vaccine that is approved for use only in a limited population, then that vaccine may not be considered adequate to address the needs of the public health emergency for other populations.
Thank you.
Mr. Tubman.
Yes. First of all, thank you and the other members of wonderful job. I think the whole country owes you a control.
You speak a little louder, please. We're having problems hearing.
Sorry. Just thanking Dr. Fick and everybody at the FDA for all the work.
Right.
Independently reviewing the data. Thank you. Two questions. One is, you indicated that since the submission came in from Pfizer, you've had a lot of ongoing discussions. And the first question is, does that include getting updated data? Because the closing date for the data that's in their submission is November 14th, just 26 days ago, which months is about half of the entire period for the 2 months minimum required for EUA. So have you gotten more recent, requested and gotten more recent data? And the second question is a follow-up to Dr. Perla. The question, you've given us only one question, and that is yes or no to recommending approval for the entire amount, which is population, which Pfizer is requesting, which is the entire 16 and over. But certainly the committee could view this as something they'd like to grant EUA for, but given the balancing of risk and benefit, it may be different for different populations, so we might want to grant it or recommend it for a smaller set than Pfizer's asking for. But your question doesn't seem to allow for that, and I was wondering if you have a backup question that might be used. I know that in the past with the dengue review, there's backup questions, so just want to ask if you can answer those.
Okay, could I say that your last question, we can bring that up during the discussion later on. So I'm going to excuse Dr. Fink from having to answer that part of the question.
Thank you.
So
to answer your first question, no, we have not received additional data sets beyond the data sets that were submitted to us comprising a cutoff date of November 14th. As you can imagine, there is a tremendous amount of work that goes into preparing a data set for submission, and so it really is infeasible for the sponsor and for FDA to be chasing our tails trying to get data sets that encompass more and more data as time goes on. That being said, if the sponsor becomes aware of, or if we become aware of any data that would potentially impact our benefit risk assessment, we do have discussions with the sponsor regarding those data.
And we will be able, Mr. Taubman, to ask questions of the sponsor after their presentation later on. So we can revisit this issue. Next, Dr. Perlman.
Yes. So I just had a question about the last part of what you were talking about when you talked about how Pfizer is going to deal with the issue of placebos going off the vaccine trial, and you asked for advice from the committee. Is that part of the EUA vote, or is that a second issue?
So that is not a question for a vote. It is just an item for discussion. We'd like to hear the committee's thoughts on the plan that is being proposed by Pfizer, which you will hear about in greater detail later today.
Dr. McGinnis.
I have a question that's sort of a follow-up from what Mike Kurilla originally started. Given that this is considered an investigational product under an EUA, I'm assuming that any manufacturer cannot actually market such an investigational vaccine. Is that correct?
So this is the emergency use authorization does not allow for commercial distribution of the vaccine via the usual marketing that would be available to a licensed vaccine. So I hope that clarifies things. If anyone else from FDA wants to chime in, I'd welcome additional remarks.
So Doran, thank you. I'm happy to get the comments. I'm just trying to understand. So it's not marketed traditionally, but compensation for an investigational vaccine is possible under an EUA, like with a government entity?
So again, this is not my area of expertise.
I understand.
The emergency use authorization may include conditions related to advertising and other similar issues in terms of transfer of money between vaccine recipients and providers, or between the U.S. government and the manufacturer. I can't speak to those.
Dr. Meisner.
Thank you, Dr. Monto. And I would like to thank Dr. Marks, Dr. Fink, and Dr. Gerber and everyone else at the FDA, because the amount of work that you have done is just extraordinary. And the briefing packets that have been circulated are clear and extremely helpful. So thank you very much for your ongoing work. The question I have for you is, it will be advantageous for a number of reasons to have one of the COVID-19 vaccines available under a biologic license application instead of an EUA. And can you, and I realize it's a difficult question, but can you offer any comments about the route or the pathway forward for the FDA to begin to think about when you've reached a point that you would consider a BLA?
Yes. First of all, we do want any vaccine that is made available under an EUA to continue its testing to allow for its licensure as soon as possible after issuance of the EUA. So this continued evaluation, as I explained in my presentation, I think first and foremost would include some longer-term follow-up of participants enrolled in ongoing studies, as well as safety and effectiveness data coming out of use of the vaccine under the EUA. This additional evaluation would then meet our usual expectations for clinical data to be included in a licensure application. There are some other sources of information that involve manufacturing and facilities and potentially non-clinical studies as well that we would typically require to support a licensure application, but are not absolutely necessary to support issuance of an EUA.
Is it possible to predict or estimate when conditions of safety and efficacy might be satisfied for BLA?
In terms of the time it would take, yeah, I couldn't predict, but I will say that we typically ask for at least 6 months of follow-up in a substantial number of clinical trial participants to constitute a safety database that would support licensure.
Thank you.
And finally, Dr. Fuller.
Overview. My question, did I understand you to say that the FDA in the October 22nd meeting looked at the manufacturing quality of the Pfizer vaccine and that was satisfactory? And if so, how will that be evaluated in the actual distribution of the vaccine? And will we talk about that later?
Right. So to clarify what I said earlier, as part of the EUA submission and in information submitted prior to the EUA, FDA has been conducting an ongoing review of manufacturing quality, consistency, and control. And we have found this information to be adequate to support emergency use authorization of the vaccine. We are not intending to discuss details of the manufacturing process, many of which are proprietary, during today's meeting. But as Dr. Gruber and I discussed in response to a previous question, our review of the manufacturing information is an ongoing process and will continue even after the vaccine is authorized, if that is the decision that we make.
Okay, thank you.
Thank you, Dr. Fink. You kicked us off with a lot of information, which we're going to be coming back to later on this afternoon. I'd like next to call on Dr. Aaron Hall from CDC, who's the co-lead in the epidemiology task force, and he is going to give us an update about the current situation.
All right, thank you very much. Good morning. Can you hear me okay?
Yes, we can.
Wonderful. So good morning. I'm Dr. Aaron Hall, co-lead of the Epidemiology Task Force in CDC's COVID-19 response and chief of the Respiratory Viruses Branch. To help frame subsequent discussions today about COVID-19 vaccines, I would like to provide a brief update on the current epidemiology of COVID-19 in the United States. As of December 8th, over 14.8 million COVID-19 cases and over 280,000 associated deaths have been reported in the United States. The initial peak in early April was driven largely by elevated activity in the New York metro area, followed by a second, larger peak in late July, primarily due to increased activity across much of the southern U.S. Since mid-September, daily counts of new cases have again been on the rise, with even sharper increases since mid-October. Since submission of these slides in advance of this meeting, we have now surpassed 15 million cases and 285,000 deaths nationally, with over 200,000 new cases and over 2,500 new deaths reported yesterday. In addition to reported cases, CDC uses several other systems to track the pandemic, which are compiled in the weekly surveillance summary called COVID View. This weekly report includes the percent positivity of molecular tests for SARS-CoV-2, the virus that causes COVID-19, shown in blue, as well as syndromic surveillance for COVID-19-like illness, or CLI, among ambulatory patients, shown in red. Both of these leading indicators have been increasing since September. Also included in COVID View are weekly hospitalization rates, shown in gray, which are currently at the highest point since the beginning of the pandemic, and also the percent of deaths due to COVID-19, influenza, or pneumonia based on death certificates, shown in green, both of which are lagging indicators that have been increasing since October. One component of the weekly COVID View report is an assessment of COVID-19 hospitalizations through COVIDNet. COVIDNet conducts hospitalization surveillance in 14 states, representing about 10% of the U.S. population. Patients must be a resident of the surveillance area and have a positive SARS-CoV-2 test within 14 days prior to or during hospitalization. Medical chart reviews are conducted by trained surveillance officers, and data are updated weekly on an interactive website. While focused on the more severe end of the illness spectrum, COVIDNet provides active, population-based surveillance, thus overcoming some of the biases with passive surveillance and providing robust data on the epidemiology of COVID-19. Looking at weekly hospitalization rates by age, we see that each of the peaks in April, late July, and currently have been most pronounced among adults aged 65 years and older. Hospitalization rates in children have been considerably lower than those among adults, but have remained stable or increasing since the spring. Note that the last few data points are subject to reporting lag and may increase subsequently. Looking at the cumulative hospitalization rates through late November from COVIDNet and stratifying by age group, we see a strong increasing trend with increasing age. As of November 28th, adults aged 65 years and older had a cumulative rate of 756 per 100,000, which is roughly equivalent to 1 in every 130 people in this age group being hospitalized with COVID-19. This rate is approximately 4.5 times greater than that of adults aged 18 to 49. COVID-19 surveillance has also helped to identify significant racial and ethnic disparities in the rates of COVID-19. Shown here are age-adjusted cumulative hospitalization rates by race and ethnicity, demonstrating rates are 3 to 4 times greater among persons that are Hispanic or Latino, non-Hispanic American Indian or Alaska Native, or non-Hispanic Black or African American, compared with those that are non-Hispanic white. These disparities are likely multifactorial, potentially influenced by differential exposure rates, prevalence of underlying medical conditions, access to care, and other socioeconomic factors. To help further tease apart these issues and identify risk factors for severe COVID-19, CDC and public health partners analyzed the relative rates of in-hospital mortality from COVID-19 using models that adjust for age, sex, race and ethnicity, smoking, and several underlying medical conditions. As shown in the red box, older age was the strongest independent risk factor for in-hospital death, and the risk increased with increasing age. Other characteristics significantly associated with in-hospital mortality include male sex, immunosuppression, renal disease, chronic lung disease, cardiovascular disease, neurologic disorder, and diabetes. Combining data from COVID-19 with population-based data from the Behavioral Risk Factor Surveillance System, or BRFSS, we likewise developed models to assess risk for COVID-19 hospitalization among adults with specific underlying conditions. Again, after adjusting for age, sex, and race and ethnicity, the risk for COVID-19 associated hospitalization was greatest for adults with severe obesity, chronic kidney disease, and diabetes, as shown in the red box. Compared with adults without these conditions, those that have them were 3 to 5 times more likely to be hospitalized for COVID-19. Furthermore, the risk of COVID-19 hospitalization increased with the number of underlying medical conditions, as shown in the red box on this table. While specific risks vary depending on the specific underlying medical condition, adults with 3 or more conditions had 5 times the risk of COVID-19 hospitalization compared to adults with no conditions. Due to increased age, underlying medical conditions, and their congregant living situation, residents of long-term care facilities have been disproportionately impacted by COVID-19. As shown here, residents of these facilities comprise nearly 50% of COVID-19 hospitalizations among adults aged 75 to 84 years, and nearly 2/3 of COVID-19 hospitalizations among adults aged 85 years and older. As such, the Advisory Committee for Immunization Practices, or ACIP, recently recommended long-term care facility residents as a priority group to receive initial doses of COVID-19 vaccines once approved. Similarly, healthcare personnel have been prioritized for vaccination to preserve capacity to care for patients with COVID-19 and other illnesses. Again, based on COVID-19 data, 6% of adults hospitalized with COVID-19 were healthcare personnel, with nursing-related occupations being most frequent. Healthcare personnel hospitalized with COVID-19 had similar prevalence of underlying medical conditions, most notably obesity, as that observed among adults hospitalized with COVID-19, all adults. Likewise, a similar proportion of severe clinical outcomes, including ICU admission, mechanical ventilation, and death, occurred among healthcare personnel as that across all adult COVID-19 hospitalizations. As we prepare for COVID-19 vaccines, it's important to establish baselines to assess their future impact and maintain ongoing assessment of the total burden of SARS-CoV-2 infections in the U.S. To that end, CDC has implemented a nationwide seroprevalence survey to help track the number of people with evidence of previous SARS-CoV-2 infection, including milder infections that do not result in care seeking or testing for acute infection. These involve biweekly testing of approximately 50,000 residual specimens from commercial laboratories for antibodies against SARS-CoV-2. Through the first few rounds of this survey, estimated seroprevalence has ranged from 0.4 to 23% across U.S. jurisdictions. However, as of late September, less than 10% of specimens from most jurisdictions had evidence of previous SARS-CoV-2 infection. In general, the highest seroprevalence was observed among children and adults aged less than 50 years, and lowest among older adults aged 65 and older. Using a different multiplier modeling approach, which uses reported cases and other data sources to then account for underdetection and underreporting, CDC recently released estimates of the total number of hospitalizations, illnesses, and infections with SARS-CoV-2 in the U.S. This analysis estimated that 1 of every 2.5 hospitalized cases and 1 of every 7.1 non-hospitalized cases may have been nationally reported. Applying these multipliers to reported cases through the end of September yielded a national estimate of 2.4 million hospitalizations, 44.8 million illnesses, and 52.9 million total infections. As the figure to the right shows, most estimated hospitalizations occurred among older adults, while most illnesses and infections were among younger adults. So in summary, now, as of December 9th, over 15 million cases and over 285,000 deaths associated with COVID-19 have been reported in the United States. However, based on seroprevalence surveys and models, the total estimated number of infections is likely 2 to 7 times greater than reported cases, though less than 10% of the population in most states had evidence of previous infection through September. Factors associated with increased risk for severe COVID-19 include older age, racial and ethnic minority group membership, and several specific underlying medical conditions. Ongoing surveillance and epidemiologic studies will help inform further development and implementation of candidate vaccines, including assessment of their impacts, safety, and effectiveness. Lastly, even with the promising advent of COVID-19 vaccines, there's continued need for non-pharmaceutical interventions, including mask use, physical distancing, hand hygiene, and environmental disinfection to help bring an end to this devastating pandemic. Finally, I would like to acknowledge the thousands of public health professionals that have worked tirelessly over the last 11 months on the CDC COVID-19 response, including the dedicated staff from the Respiratory Viruses Branch. Thank you.
Of COVID, I think I wasn't on at first. Thank you so much for being clear about the differential impact of COVID-19 in the U.S. population. We have time for relatively few questions. I see Dr. Meisner. Do you have your hand raised?
Yes, sir. Thank you very much for that presentation, Dr. Holmes. I would like to ask you about the severity of disease, particularly in older adolescents, that is, individuals who are 16 and 17 years of age, because they have been included in the company's request for an EUA. I assume it's unlikely that hospitalizations and disease are broken down by age, but is it safe to assume that adolescents who are 16 and 17 years of age are similar in the 5 through 17-year-old age group?
Yeah, thank you for that question. So, of course, as we refine to smaller and smaller age brackets, the numbers get smaller, particularly in children, where overall we see lower rates, particularly of severe disease, of hospitalization. In general, we do see higher rates of hospitalization among children aged 0 to 5 relative to those aged 5 to 17. Of course, this is potentially confounded by differential rates of care seeking. As you might imagine, there are certainly lower thresholds for care seeking for the very youngest and most vulnerable children. However, as we start to look at the more mild end of the illness spectrum and look at rates of detection of SARS-CoV-2 virus using molecular assays, we do see indication of higher rates of infection among older children, among adolescents, particularly then as we move into kind of older teenagers and people in their young 20s. Much of this may have been driven in part by outbreaks in the fall among institutes of higher education, but the broader impacts in children perhaps are not entirely clear until the full resumption of normal activities ensues in the United States, including in-person education across all schools in the United States. So we'll continue to monitor it closely, but thus far the indication is the highest rates of illness, severe illness in young children, but higher rates of infection in older children.
Thank you.
Dr. Rubin.
Thanks, Dr. Hall. I was interested in the multiplier that you described. That applies to the diagnosis of infection. Do you think that applies to deaths as well?
Yeah, so we have used at CDC the same multiplier model previously for tracking influenza and generating in-season estimates of the disease burden. And that same approach has been used previously to estimate deaths in the same manner that I presented today for hospitalizations and milder illness. There are, of course, differential multipliers that would have to be considered for deaths, as the rates of underreporting of death are different than those for milder infection. In general, we have better capture of deaths. We have lower rates of underreporting and underascertainment for deaths. But as folks are aware, the dynamics of the pandemic itself have greatly changed those multipliers. And so there's also a considerable time component that needs to be factored in when using these multiplier models. So the underreporting of deaths, for example, has changed over time, and the attribution of deaths to COVID-19 has changed over time, which complicates interpretation. But we do have several efforts underway using these models and other approaches to generate estimates of death. And we do feel, as with hospitalizations and illnesses, that the reported number of deaths is likely an underestimate of the true number of deaths.
Thank you.
Thank you. Dr. Gans, we're going to go over a little bit, but please keep your questions short.
Thank you very much. Thank you for that, and I agree, heroic effort to all of the people who are working on this. I had two quick questions. The data concerning the immunocompromised or immunosuppressed individuals is not really granular enough to make it something that we can use in terms of how we're thinking about high-risk populations. There are registries looking at this, but it would be nice if there were some national information that you could provide to us, as well as I didn't see any information from pregnant women, which is a population which we're all concerned about, and we've definitely seen postnatal transmission to very young infants who end up being hospitalized. If you would just take those up.
Yeah, thank you for those comments. Absolutely. There are numerous surveillance efforts currently underway to assess the impacts of COVID-19 in pregnant women. In the interest of time, unfortunately today, I didn't have a chance to present those, but through COVIDNet, as described today, as well as another surveillance system called SetNet, which was established during the Zika epidemic. We have been very closely monitoring the impacts of COVID-19 in both pregnant women and subsequently following up with their infants. Thankfully, the rates overall have been relatively low thus far, but as we continue to accumulate a critical mass of data in this demographic, we indeed do hope to have more specific estimates of the risks that are posed to pregnant women and their infants. The early indication is that there may be a higher risk of preterm delivery among pregnant women infected with COVID-19 relative to women without COVID-19. But there's ongoing efforts to assess those and other potential pregnancy-related risks and fetal outcomes.
Thank you very much, Dr. Hall. And it's now time for our morning break. We've eaten into the time a little bit, but I do want to start again at 10:30 Eastern, so we can hear from Dr. Messonnier from CDC. See you later.
All right, welcome back from break. We'll now enter our second half of our next group of speakers. Arnold?
We're next going to hear again from the Centers for Disease Control. We're going to have a few presentations and questions afterwards. First, we're going to hear a presentation from Dr. Nancy Messonnier, the Director of the Center for Immunization and Respiratory Diseases, on vaccine safety and effectiveness monitoring. And then from Anita Patel, the Deputy of the Vaccine Task Force on Operational Distribution Plans. Dr. Messonnier.
Thank you, Dr. Monto, and thank you for the opportunity to speak today on this really auspicious day. Despite the completely appropriate size and scope of the clinical trials, it's always important to continue to monitor vaccines post-licensure, post-authorization, and that is certainly especially true with COVID vaccines. So I'm going to talk about the U.S. government plans for monitoring of safety and effectiveness. First, safety. The rapid implementation of safety monitoring under an EUA requires a whole of U.S. government approach with an initial focus on the early populations targeted for vaccination. Voluntary active surveillance of adverse events focused on healthcare worker vaccination and rapid follow-up of reported serious adverse events. As the program continues and more vaccine is given, active surveillance systems will provide increasingly useful information on safety in different populations. Close collaboration of safety experts across the USG will facilitate data sharing and rapid recognition of responses to safety signals. Now, there is a quite extensive plan for safety monitoring, but I'm just going to summarize a few high points. On day one of the COVID-19 vaccine programs, systems will be in place to monitor the safety of vaccine recipients. These include the traditional systems that we use, VAERS, which is a National Spontaneous Reporting System monitored and implemented by CDC and FDA, but also similar programs at DoD and the VA, as well as the National Healthcare Safety Network, or NHSN, which is an acute care and long-term care facility monitoring system. CDC CSIS system is stood up that provides individual case consultation. But in addition, to really enhance this period, CDC has stood up a new system called vSafe. It's an active surveillance tool that uses text messaging and web-based surveys to monitor vaccine recipients for adverse events. Recipients complete brief surveys up to really intermittent surveys to 12 months following vaccination. But then as part of the program, recipients that report a significant health event that impacts their daily activities or leads them to seek medical attention will be called for more information and a report will be generated. This is an opt-in system, and so we're going to have a heavily enhanced engagement around implementation of the program. We really need people to sign on to this system to give us the best data possible. Oops. Large linked database monitoring systems will provide safety data when vaccine becomes more widely available in the priority groups and the general public. This includes, again, existing systems like VSD, the FDA's extensive system, and systems from DoD and VA. We're enhancing this by Genesis, which is a new collaboration with NIH and Brown University to monitor safety in long-term care facility residents. This system operates nationally and has near real-time data for 250 facilities. To look at this another way, this is a table that showcases the systems that we'll be using to monitor safety in the populations that, based on ACIP recommendations, will be vaccinated first, specifically healthcare workers and long-term care facility residents. So you'll see that early on, we have a series of systems to monitor healthcare workers and long-term care facility residents through VAERS. VAERS will be enhanced in this time period especially around long-term care facility residents, to ensure that we're getting those reports. And then Be Safe, which will specifically target healthcare workers. And then you'll see all the systems that are going to be in place for later monitoring in all of those populations. I think the team has done a really good job of thinking through what systems we have, how to enhance those systems, and looking for any weaknesses and attempting to fill them in. And, you know, I'm giving it short shrift by going through it so quickly, but hopefully you'll understand how robust these systems are. In addition, the ACIP COVID vaccine safety technical subgroup was established to provide expert consultation on COVID-19 vaccine safety issues. This group, which we call VAST, was built off lessons learned during H1N1 safety monitoring. The terms of reference and composition have been finalized, and VAST is ready to begin reviewing data once implementation commences. The group is co-chaired by a member of ACIP and a member of the National Vaccine Advisory Committee, or NVAC. There are 10 independent expert consultants that make up the group, as well as ex officio members from NIH, FDA, OIDP, CMS, HRSA, and IHS, and we also have representatives from the VA and the DoD. Now, to briefly summarize our efforts around post-authorization vaccine effectiveness. Post-authorization or post-licensure VE estimates are needed to address important evidence gaps from phase 3 clinical trials, particularly for secondary endpoints that may not be adequately addressed. For example, the trials may be limited in their ability to address VE against secondary endpoints like infection transmission or in subpopulations, and we won't have great data early on the duration of protection. We also believe it's important to evaluate the real-world performance of vaccines, as protection may differ from efficacy under trial conditions. So, this table attempts to lay out the VE policy priorities that were determined based on consultants with policy experts and internal and external experts. The most immediate priority is to determine whether the vaccine protects against symptomatic disease as expected based on the phase 3 trial data, but understanding that implementation for these vaccines might be slightly complicated by folks who don't exactly get the second dose exactly on schedule and by the necessity to keep the vaccine in cold chain. So that's going to be the focus, but then there are a variety of subsequent and late stage studies that are being planned or implemented. A number of strategies are used to plan for the assessment of VE, but to facilitate a rapid launch, we've leveraged existing platforms, and for early stage vaccination, we focus on populations likely to be vaccinated first. We're harmonizing and coordinating across the U.S. government on a variety of platforms, for example, using common case definitions, data elements, and methods to improve comparability of results. And we've been working to combine similar platforms to improve geographic representation, increase statistical power, and generate more timely and robust VE estimates. And we do have a diversity of methods as we understand that all observation methods have limitations. Here's a summary of the studies that are being planned. And again, I don't have time to go through all of them, but I would just put you to the top row. You know, for the most immediate question of whether vaccines work as expected, we're planning a prospective study among healthcare workers. There is no large existing database enriched for this group, and so we're doing a test negative design case control study among healthcare workers, which should help us relatively quickly assess this outcome. So, in summary, planned VE studies will provide a robust assessment of COVID-19 vaccine performance in real-world settings. We believe we will have early VE estimates in groups prioritized for vaccination, VE against key outcomes, and in important subgroups. And data from these studies will address critical gaps and help guide future use of vaccine. Systems are in place to monitor the safety of COVID-19 vaccination in healthcare workers and long-term care facility residents. BSAFE, VAERS, and HSN will detect adverse event signals for further follow-up and evaluation. CMS, Genesis, and other claims-based and EHR systems will be used for both signal detection and evaluation. Vaccine effectiveness in healthcare workers is the immediate priority and will address the question of does the vaccine work as expected. We expect those studies to start immediately. And vaccine effectiveness evaluations in older adults, including those in long-term care facilities, are planned and include both test negative design and cohort evaluations. I hope that does justice to all the systems that we're planning and that are in place. Understand that vaccine safety and effective monitoring is a top priority for the U.S. government, and we're committed to ensuring that all these systems are in place and ready to go as soon as the vaccine program is implemented. Thank you.
Thank you, Dr. Messonnier. We're going to go directly on to Dr. Patel's presentation, and then we'll have questions afterwards, so we have plenty of time. Dr. Patel.
Great, thank you so much. So thank you for having me today. And we'll be walking through today at a high level, what the USG's distribution strategy is for COVID vaccines, specifically focusing around the Pfizer vaccine product. The USG plan for COVID vaccine distribution will adjust as volume of vaccine doses increases. Early on, we do anticipate that we'll have a constrained supply where vaccine administration is really targeted towards select population groups, and distribution will facilitate that. As sufficient supply becomes available and the populations that we're trying to reach expand, our distribution network will also further expand. The schematic here outlines what our concept is for distribution at a very high level. All vaccines will be ordered through CDC's COVID vaccine ordering systems. For the Pfizer vaccine, we will be providing those vaccine orders directly to the manufacturer for distribution to administration sites. We've been working very closely with our state and local partners to make sure that they have plans in place to identify providers that are able to receive, store, and use the vaccine for the specific populations that they're trying to reach. These administration sites will be receiving the Pfizer vaccine product, and they should expand as well over time as the targeted populations we're trying to reach expand. In addition to the hospitals, the vaccination clinics that have been set up to support early distribution and use of Pfizer vaccine, we have also set up a program called the Long-Term Care Facility Pharmacy Program. This federal program is a partnership that allows for jurisdictions to be able to opt into the program, allowing for on-site vaccination support to occur to long-term care facilities. Our pharmacy partners would be providing the on-site vaccination support for those facilities that actually sign up for the program. Also like to draw your attention to the Operation Warp Speed Coordination Cell at the bottom of the slide. The idea is for us to be able to have a clear understanding of what is happening in terms of supply of vaccine as well as administration and uptake information throughout the vaccination program. Focusing a little bit more on the long-term care facility plan, in order for us to be able to reach skilled nursing facilities, we have been working to bring skilled nursing facilities on board to the program and opt in. Once a state determines that they're ready to expand vaccination to reach staff and residents within skilled nursing facilities and other assisted living facilities, the program would be turned on. Right now, we do have a very high percentage of the skilled nursing facilities within the United States that have opted in to receive services from this federal program. The vaccine characteristics of the Pfizer program are very different and unique compared to other vaccines that we've seen before, and they do impact distribution, site storage, as well as administration requirements. The details depicted on the slide are very specific to the Pfizer program and outline some of the challenging requirements of the vaccine that have to be implemented in terms of vaccine distribution and storage sites. The additional planning that is needed for distribution, storage, handle, and use has been underway for the last few months based on the information that we know for the Pfizer product. There is a required dedicated cold chain given that the product has to be managed at ultra-low temperature requirements. In addition, the timing of the vaccine ancillary supplies, which include the diluent, as well as dry ice replenishment, has also been a challenge. We have built the distribution system in a way that all three of these elements will arrive within a 36-hour period of time. So once the vaccine is there, these additional pieces will also be delivered to the vaccination sites. Another major challenge that we've had with the product is the actual minimal order requirement. 975 doses is the smallest volume of product that can be ordered for the Pfizer vaccine. This has posed challenges, especially in rural areas of the country where that volume of product is more difficult to manage. And it's also been a planning factor that at the local level has been used to determine where exactly the product should be received. There's also a new element of the thermal shipping container that has been introduced with this product. The container allows for the movement and shipment of the vaccine from one site to another and allows it to be managed in the ultra-low temp conditions. There is training required with using this thermal shipping container and staff being able to be trained on the monitoring, the dry ice replenishment requirements, as well as understanding how to manage the product over time, how many times to open the container, and the details have also been part of the training and what we're getting vaccination sites ready for. There's also complexities with mixing and using the product. It is a little bit different than other vaccines that we've seen as far as the diluent being required, how the product is drawn up, and also how quickly the product needs to be used. Once diluted, we do have a 6-hour period of time in which the vaccine needs to be administered in, and that has also been factored in and built into the planning at the administration site level. Lastly, we do have inventory planning that is underway to ensure that there is sufficient supply available for administration. This includes ensuring that administration sites have actual clinic planning times, that they're able to have enough people come to manage the 975 doses for each of the trays, and the planning for the actual clinics, as well as the planning for the second dose. So second doses are required, the timing, 21 days, and any cushion that we have beginning or ending from the 19 to 23 days that we saw in the clinical trials. What exactly that looks like and how that gets operationalized and implemented is also part of the planning factors. Second dose reminders are part of ensuring that people come back for that second dose, and additional strategies are also underway. So with that, I'd like to pause and turn it back over to the moderator.
Thank you, Dr. Patel. We're going to have time for just a couple of questions at this point. Dr. Levy.
Hello. I have a brief question for Dr. Messonnier. First of all, thank you so much for your presentation. It was very helpful. You needed to move very quickly through some very important information about the safety surveillance systems that are in place to monitor the progress of coronavirus vaccine. I did have a question. You described a number of interlocking national systems that would mirror vaccine safety. I wanted to know if there's any consideration or plans for also integrating these systems with international efforts at monitoring coronavirus vaccine safety. For example, World Health Organization, WHO. There's also an international network of special immunization services and others. So that's my first question. My second question has to do with vaccine effectiveness. You mentioned some key subgroups that you'll be monitoring vaccine effectiveness in. Those all made good sense. It went by quickly, but I didn't see teens or maybe even eventually younger children listed among those special populations, but I would imagine they would be of interest as well, and I'd value your comments on that. Thank you.
Thank you for easy questions. The answer to both questions is yes. There's actually a WHO coordinated group looking at sharing data for both safety and effectiveness and coordinating across the globe on that. In addition, we are working directly with collaborators in the UK and in Canada to make sure that we're sharing information as quickly as possible. And we're already gearing up to help a number of countries that are for immunization campaigns. The answer to the second is definitely yes. I didn't put it on the slide because authorization for those groups was not really up for discussion today, but certainly it will be really important to be able to monitor both safety and effectiveness in an enhanced way in those groups. Yeah, period.
And the final question from Dr. Kurilla. After that, we'll have to move on.
Thank you. This is for Dr. Messonnier. You described a very comprehensive assessment, aggregation, and collation of a lot of safety signals, and I'm wondering, the intention for that group, is that to provide ongoing advice to the people who would be distributing the vaccine, potentially administering it, public health officials, or is that primarily to be focused towards the FDA in terms of refinement of the EUA itself? What's the real focus of all of that safety assessment? Where's it going?
I mean, I think it can be for multiple uses. However, our standing up of an ACIP work group is to be a part of our ACIP recommendations, that is the recommendations for use of the vaccine. CDC and FDA and all the USG partners coordinate, collaborate, work together in the safety landscape every day on all our routine vaccines. And as you look at our systems, you'll see how intertwined they are. So any information, for example, that we get will be shared quickly with the FDA, as well as putting these systems in place so that our advisory committee can review it. I also might say that CDC and FDA are also discussing a contract with NASEM, the National Academy, so that we also have them to provide expert independent review of vaccine safety issues that may arise in the COVID-19 vaccination program. So in addition to everything we've set up, we're also setting up that collaboration contract with NASEM so that we also have them ready if we need to help adjudicate a safety signal. Thank you.
Thank you.
Well, thank you to both of our CDC.
Thanks for taking me to this advisory committee today on one of its most important decisions, and the title of on this slide is exactly what I'm speaking to. There we go. So what are these considerations? Well, there are two, and they're intertwined. First are the ethical considerations, which concern the questions about what is the right thing to do with the placebo group in this case. The second class of considerations are epistemic, concerning what we know or what we believe, which is determined by the strength and totality of the evidence. That's what's generated by the designs we are already using and the ones we'll use going forward, which might be the most consequential thing that we discuss today. Now, I will be speaking about ethical issues, but I am not a card-carrying or PhD-carrying bioethicist. But I've enjoyed working with many, and I want to acknowledge, before I talk, the contribution several have made to my remarks today, although the views and ethical lapses are my own. They are Professors Nancy Cass and Ruth Baden from Johns Hopkins and David Magnus from Stanford. Now, there are a lot of questions embedded in what should happen with the designs and particularly the control groups in studies going forward. This slide outlines a lot of sub-questions, but I think the title of the presentation pretty much captures it. If an EUA for this and other vaccines are granted, what do we do with the placebo control groups in this trial and potentially in ongoing and in upcoming trials for the next vaccine? Now, I want to start with a set of ethical preliminaries. I'll start with something that seems very basic, but I can't tell you how many committees I've been a part of and that I've watched whose conversations have founded on this simple issue. We're going to be dealing with ethical dilemmas here, but it's worth thinking about what an ethical dilemma is. So an ethical dilemma is not a choice between right and wrong. That's what we write to advice columnists for. An ethical dilemma is a choice between two actions that are both ethically justifiable under different moral frameworks or principles. In that sense, it's a competition between two right actions, which of course is exactly what makes it a dilemma. We face this in every clinical trial, particularly on data monitoring committees, the tension between actions that are good for an individual, which in the current setting might mean giving them a vaccine immediately, and what's best for society, which might mean holding off on that vaccine so placebo-controlled randomized trial can continue. Resolution of ethical dilemmas always requires compromise that balances the competing dictates and minimizes physical harms or ethical wrongs which are caused by unfairness, violations of autonomy, or actual personal or societal harm. Now, to continue, our ability to conduct clinical trials, which is to experiment on each other, is something that society gives us permission to do. We don't need that permission to do chemistry or biology experiments. And because the procedures and oversight for RCTs are so structured and standardized, it's easy to forget how fragile that permission to conduct RCTs is. But anybody who's worked as an institution whose research enterprise has been completely shut down because of a single ethical lapse knows that this trust can never be taken for granted. And it's continually earned and re-earned through fair and transparent processes to determine what's acceptable. And that's what we're doing today and why it's so important that we're doing it in public. Now, final preliminaries, which is ethical acceptability depends often on context or background conditions. Offering a placebo subject the vaccine can be judged quite differently depending on whether the evidence, we have about efficacy and safety, and whether or not that participant can actually get the vaccine outside of the trial. And both of those are part of the context today. Now, epistemic preliminaries. I'll start by saying something that shouldn't be too controversial, which is that RCTs, or randomized controlled trials, provide the most reliable knowledge about the relative efficacy and safety of interventions, at least as permitted by the sample size and follow-up time. Obviously, we can't detect safety issues that come up after the RCT closes. A second principle is that reliable knowledge can come from non-randomized designs, and Dr. Messonnier just introduced us to some of those. This includes exactly the kinds of things she described: vaccine safety surveillance, all of epidemiology, quasi-randomized designs, natural experiments, and understanding of biologic mechanisms. These can play a critical role. And finally, there are many things to learn about these vaccines. We've talked about some already. Some of them are best learned through an RCT, but not all of them. And there are many things we can learn from these other designs that do not require RCTs. Now, finally, in this stage setting, there should be no bright lines. That is, bright lines are almost always unjustified, and they are the enemy of ethical and epistemic compromise. In ethics, this means we can't allow one moral framework or principle to trump another. We can argue about how one principle takes precedence over another, but it's not helpful to hear assertions like, it's unethical to deny an effective vaccine to a placebo recipient. Asserting that anything is unethical effectively shuts down the debate, and it also demonizes people on the other side. And I found it extremely healthy for committees not to use that word at all. There's a corollary in epistemology where we can have reasonable discussions about what we can learn and what we can't learn from randomized and non-randomized studies, but saying things like you can't learn anything from a study without randomization is both obviously not true and inimical to a balanced solution. Now, what do RCT participants know? Let's get into the issue of ethical obligations. All consent forms and consent processes are required to tell the patient or the participant in the study that the purpose is to produce scientific knowledge that benefits society, not necessarily them directly. They're also told that they trust that they won't be exposed to known serious harms, not listed in the consent, and that they can withdraw at any time for any reason, and that they'll be given information arising during the trial that's highly relevant to their decision to continue. This is exactly what is in the Pfizer consent. In quite plain language, I've put it up here. I won't take the time to read through it, but it's quite explicit, and we have no reason to believe that the study was misrepresented in any way. Here in the highlighted area, you can see you're free to stop participating at any time, and the study team will tell you in a timely manner if new information is learned that would change your mind about continuing that study, and that certainly would be an EUA. Now let's talk about, I'm sorry, skip the slide there, whether there's special obligations to placebo recipients. Obviously they have exactly the same, we have the same obligation to them that we have overall to the trial. They're free to withdraw. And because of that, the trial structure investigators shouldn't actively deny them vaccine in any way if it became otherwise available to them through a social prioritization and local circumstances. And this is consistent with the FDA position stated earlier. We'll come back to this. I would also argue that the investigators are not obligated to provide immediate vaccination in the trial before their turn of the participant is called outside the trial. First, this is not what they signed up for. And you know, there are many prioritization criteria out there by very, very good groups. And I haven't seen one that includes trial participation as a basis for jumping the queue. That is not considered a reason to get it before somebody else who might deserve it or need it because of higher risk or other considerations. But it should be reasonable when a trial participant becomes eligible through societal priorities and local ability, that they then can be provided vaccine in the trial context. So in a sense, they jump to the front of their own line. And that also binds them to the trial. It's a form of reciprocity, and it facilitates further follow-up. Now, what are some issues related to the COVID-19 context that will have relevance to these decisions? The first is the most obvious. We're in a pandemic. It's a public health emergency, which magnifies potential consequences, both positive and negative, and knowledge or ignorance and the consequences of knowledge or ignorance and of action or inaction. I would say that some rules that apply in peacetime might have to be modified in war, and we're in a bit of a war right now. But I want to focus specifically on the health risks to individuals. Given the recent numbers, not yesterday's, which are particularly terrible with over 3,000 reported dying, but those in the last few weeks, a randomly chosen U.S. citizen has an average risk of dying from COVID in the next 6 months, that is by the end of May, of roughly 1 in 1,000, and the risk of hospitalization of roughly about 1 in 200. These numbers obviously vary widely by individual, and they don't include the harms from non-fatal COVID, but they're highly ethically relevant, and let's start to talk about that. So if the maximum, if the risk of death in the U.S. in the next 6 months is around 1 in 1,000, that means the maximum benefit of the vaccine for an individual, at least with respect to mortality, is also 1 in 1,000. So, or 0.1%. So imagine if we had a drug that purportedly raised cancer survival from 40% to 40.1%. If there was more to learn, and we do have that here, that's a setting where we would typically allow many placebo-controlled trials. We see this in meta-analyses all the time with multiple trials, placebo-controlled trials of agents with therapeutic benefit orders of magnitude larger than this. So while this risk obviously has huge population impact, in terms of trial ethics, it's not the kind of risk that incurs a great ethical debt to the placebo group or a prohibition on further placebo-controlled trials. Also, unlike a therapeutic drug for patients with a disease who can't change their own prognosis, many, but not all individuals can take actions that reduce their own risk of death from COVID, like masks and social distancing. So as long as there's still important things to learn about the vaccine, placebo-controlled trials should not be regarded as unethical. However, a big however, they might be infeasible. And that is a big issue because people may not be willing to either remain in the study or to enroll. But it's very important to not conflate the feasibility with the ethics. Now, in terms of the vaccine context, what we know, I'm actually not going to spend my time on this. You're going to hear, you've already heard a lot about the Pfizer vaccine. The only one I want to highlight is that if a participant wants to unblind themselves in the trial, they can, if they have the resources and knowledge about which antibody test, commercial antibody tests, would unblind them. Some of them are to the spike protein, which would unblind them. Others are to the nuclear capsid, which might not. Now, in terms of the vaccine context or background conditions, what we don't know, well, this has actually already been covered. There's a whole raft of things that we don't know yet and that we want to learn, both from continued RCTs and also from observational studies. Now, I want to outline a possible adverse scenario. If placebo participants who want vaccine when otherwise available and eligible are not given access within the trial in the name of scientific rigor, there may be many who respond to appeals by investigators not to seek vaccine, even if available to them. And to them, this argument doesn't apply. And we've seen that willingness to hang in studies with placebo controls in many other trials of even greater personal consequence. So it's not at all unlikely that there would be a sizable number of people who wouldn't do that on appeal. But some participants at higher risk will want the vaccine as soon as it becomes available to them. And if they can't get it through trial mechanisms, many will lose trust in the investigator in the trial itself, may leave the trial without further follow-up. They may seek vaccine outside the trial. And as more vaccines come on the market, perhaps they'll get another one that's available regionally with very uncertain effect. This will get a lot of publicity, meaning that recruitment to future vaccine trials will be that much more difficult because of a collective loss of trust. So some might appeal to the experimental status of the vaccine under the EUA as justification, as ethical justification for maintaining a placebo control. I understand why this has salience within the FDA, but I suspect that this distinction will not get much traction among future or current or future trial participants when literally millions of people are being urged to get vaccinated as quickly as possible. The point is that once the vaccine is widely available, or any vaccine, widely available and encouraged, the ability to maintain double-blinded placebo-controlled groups voluntarily for more than a nominal period, by which I might mean perhaps a few months, will likely not be under the control of the sponsor of the FDA. And some efforts to do so could undermine future trials as people don't trust that their interests will be watched out for. Now, I want to mention an important countervailing view, which is that many people out there, as we well know, are very wary about an expedited approval process and refuse a vaccine that they believe to be under-tested. They want to see those trials going to their planned end, but unless these people can be found and are willing to enroll in placebo-controlled trials, the argument about the feasibility of the trial still holds. Now, what's the Pfizer proposal for continuation? Now, it implicitly recognizes this reality, and it's consistent with everything I've discussed so far, except in one important regard. First, they propose urging as many people in the trial to stay as long as possible, which I think is the right thing to do. For placebo recipients who become eligible according to local or national recommendations, that's an important condition, and who ask for the vaccine, they propose unblinding them and vaccinating. For those who stay in the trial blinded, it's proposed that everyone be vaccinated after 6 months and followed for an additional 18 months to the trial's planned end. This is coupled with a robust observational pharmacovigilance plan for long-term safety monitoring that augments what the FDA and CDC are doing. While not unreasonable, I think that we have to remember that this design will not only unblind placebo recipients, will also unblind vaccinated participants who make the same request, because of course they won't know. And once unblinded, risk behavior can change. Many of the benefits of randomization within the trial are lost. So is there an alternative? And I would say there is and should be considered. There are two alternative designs that achieve the same ethical goals without compromising without compromising on the epistemic ones. That is, we can give people vaccine who are eligible and who want it, but maintain the blinding, thereby preserving trust that the investigators and regulators care both about the science and the participants. And let's talk about those designs. One is called deferred immunization. It's been presented in a variety of settings and gotten coverage. Also a blinded crossover design. And the second are active control designs, which are really not the issue for today, but I'll discuss them briefly. The first of these was developed by a biostatistician at the NIAID, the NIH, named Dean Follman. It involves blinded crossover designs. And the idea here is that the placebo, when either a placebo or vaccine recipient becomes eligible for a vaccine or requests it, exactly the situation that the Pfizer proposal describes, that the placebo recipient get the vaccine and the vaccine recipient get the placebo without being told what they were originally. So the blind is maintained. And then they are followed forward. So they stay within the trial confines and to some extent also protected by the randomization. As I mentioned, the second design is active control. At some point in the evolution of vaccine testing, we may no longer be able to conduct placebo-controlled trials of any type in settings where vaccines are available. Although it's not a question for today, at that time, when there are a lot out there, a comparison vaccine with reasonably well-established properties may have to be used in one arm in those areas where they're available. So we're not at that stage yet, but we may be within the next year in some countries. Now, in countries or regions where vaccines have seen poor distribution, it may still be possible to conduct placebo randomized trials, but that has ethical complications of its own, and I'm not going to get into that here. I want to talk just a little bit more about the deferred randomization scheme, and you see that in this picture. The idea is that we start with the vaccine and placebo arm as we already have, and then at the point when people are eligible and ask for the vaccine, you have to have both conditions. That's the point where a blinded crossover occurs. Because every participant is ultimately assigned to the vaccine arm at some point, this design actually, in some situations, in very plausible situations, can have greater or equal power to detect the duration, the sustenance of immunity, more than a standard 50-50 RCT. And you can sort of see that in the figure where you see the contrast between the vaccine efficacy in the width of the bar in the delayed vaccine arm compared to the waning efficacy in the original arm. And this increases over time, and these can be compared statistically. So you can see how this can work for vaccine efficacy. But of course, it decreases power to detect some other things like long-term safety issues and some other important parameters, but it doesn't lose them completely, as completely as designs where subjects are unblinded and effective randomization is lost. So if the standard 50-50 RCT is not reasonable or feasible, which I believe it very quickly won't be, this design is far better than any that would break the blind and break the randomization. We can, of course, also reduce some of the ethical tension in any in placebo-controlled trials by increasing the allocation ratio to the vaccine arm, but that may not be enough to solve the enrollment problem. The next slide shows another picture of this deferred vaccination schema, and it shows that it's possible. You see the crossover on the right. On the left-hand side, we have the trials as they've been designed so far. On the right-hand side, you see what they look like after the crossover, which is that everybody's been immunized. But it's possible statistically to impute an unvaccinated control group to help estimate secular trends. So to finish up, I want to summarize that these alternative designs require compromises, the kind of compromises I talked about before, on both the ethical side and the epistemic side, that is the evidence. Ethically, placebo recipients don't get a vaccine as quickly as some might want, and this will become more of an issue going forward as new vaccines need to be tested with multiple ones already available through EUAs. But of course, those subjects are not being forced to enroll. But the ones who do enroll can expect to get immunized at a certain number of months going forward. As noted, placebo-controlled trials may continue to be possible in areas or populations for whom vaccines are not available, but that is complicated for other reasons. Finally, on the epistemic side, with this crossover designs, we don't learn as reliably or as quickly about long-term safety and infectiousness and other factors, although we still might learn as much or more about durability when compared with a trial that continues with the placebo arm. But as I said, that trial may not be doable. We might not have the perfect design going forward, and continued blinded observation of the deferred vaccination group can still be tremendously informative and potentially more informative than one with broken randomization, particularly in combination with observational studies of the population that we've heard about today. By reducing the ethical tension within the trial and by retaining the trust in trial testing system, it provides a way further for future studies of other vaccines. So none of these are the last words on this very difficult and complicated subject, but for today these will be my last words. So I hope this will be helpful to you, and I welcome your questions and comments. Thank you.
Thank you, Dr. Goodman. You've given us a lot to think about and to discuss. I just wanted to start by raising one question. With the blinded crossover design, when would that take place? When an individual becomes reaches the priority listing for their group, or would that be at the time of issuance of the EUA?
So this is a subject of tremendous controversy and discussion. For the reasons I've already outlined, I don't think it should be, and this is my opinion, at the moment of the EUA. I think because otherwise they are jumping the queue, and I discussed that earlier. I believe it should be when for those subjects, they come up on the social they come up as they would come up in terms of priorities for the vaccine, and it's available to them in their area. That is, they can get it outside the trial. As soon as they can get it outside the trial, then this would kick in, because we want to keep them in the trial. If it's given at another time, we're basically giving them priority over other populations that have been deemed to be at greater risk or greater need. And the price I think we have to pay by compromising the trial, I believe would be too high, and we don't have, again, I made this argument earlier, I don't think we have that special obligation to give them the vaccine immediately. Those are my views.
Okay, thank you. I think we'll defer questions about how feasible this is to the sponsor after the sponsor's presentation. Next, yep. Dr. Levy. And please unmute and show yourself if you'd like.
Sorry, that was an error. I don't have a question.
Okay, Dr. Meisner.
Go ahead, Dr. Meisner.
I guess I answered all questions.
All right. Should we go to the next one?
Yeah, Dr. Chatterjee.
Yes, thank you, Dr. Goodman, for your presentation. It was certainly thought-provoking. I have a question with regard to the current study participants. By reviewing the documents, the briefing documents, it appears that there's a difference between participants who received the active vaccine versus placebo recipients in terms of adverse events that they experienced. And so there's a potential that people who are participating in the trial can actually, guess which arm they were randomized to.
Absolutely, yes.
Yeah, so is there then a possibility that those who believe that they were placebo recipients might wish to withdraw in greater numbers, and that would then compromise the study as well?
Again, it absolutely could. So even going forward, it could, and that would be an issue for the statisticians and for the FDA. The point of this particular proposal is this would give them an avenue not to withdraw. That is, stay within the trial under these conditions. Now, if they didn't want to wait for their allocation to become available and their eligibility locally, withdrawing wouldn't do them anything. It wouldn't get them the vaccine any earlier. So there would be no motivation to withdraw. And also, I do think that the majority of people who signed up for these trials did so in good faith and with altruistic motives. I do believe that the vast majority want their contribution to produce a valid scientific result. So I don't think self-interest completely in any way dominates. And again, yes, I do believe that there's a fair amount of unblinding just due to reactogenicity of the vaccine, and obviously non-reactogenicity of the placebo. But this, again, at the time when they might be able to get the vaccine outside the trial, this would give them a way to still contribute to the science of the trial without leaving. I don't know if that answered your question.
Yeah, no, that did, and I was thinking about the people who would meet the priority criteria. Obviously, they must be the ones who would want to withdraw if they thought they were in the placebo arm.
Correct, and I do believe that's what I said. I do believe that could happen.
We've got a lot of questions. I think and what we're going to do is I'm going to ask all the speakers to please be available during our discussion, because we may have additional questions that come on up at that point. I'd like to move on to Dr. Kurilla, please.
Thank you, Arnold. A great presentation. So the blinded crossover design looks quite feasible. However, the caveat is that it does take longer to get to the answer. Did I hear that correctly? That's number one. But would we also be facing a first mover effect where this would basically dictate future trial design short of any vaccine having a full BLA going forward? So everyone else will have to consider this option as a de facto approach.
Yeah, that's a really good question. That's why I emphasized that. I'm sorry, I see you talking, but I don't hear you. I'll just answer what I heard. I can't judge exactly what will happen in the future because fact patterns are going to change. So I hesitate to predict too much. It is possible that this might represent, as we go forward, a default design that reduces the ethical tension of these trials as vaccines, as I mentioned, become available in the area of potential enrollees in the trials. And what we then be asking them to do is not completely deny themselves a vaccine, which they could just go out and get if they themselves are eligible, but just defer it for a few months. It might be 2 months, 3 months, 4 months, and that might be less of an ask. I can't say whether it will become the default. Again, it's going to be a while before enough vaccines of any type, no less just this one, are rolled out, even in the U.S. Remember also that these trials are going on all over the world. So the U.S. Places where they will not always be occurring in places where vaccines are otherwise available for quite a while. So it is possible we might evolve. I mean, I do see that there's going to be a staging where there's going to be a point or places where we cannot any longer do the placebo-controlled randomized trial. We might move to this, and then ultimately, as I said, and it might be a year or two from now, have to move to active controls. That's the evolution I see. Whether you're committing to this design going forward, harder to say. That's very complicated. And again, it's going to depend a lot on vaccine distribution and what we learn about these vaccines. If there start to be safety signals that we're not seeing right now, that could change the dynamic dramatically.
Thank you.
I'm just going to allow one more question because we have to start at noon, which is difficult to. So, Dr. will be the last question, and we're discussion as part of. We'll really want to pick where we've left off on some of these issues at that point. So, Mr. Tubman.
Yes, thank you. I really appreciated the presentation. On the blinded crossover design, so I understand this. Basically, you solve the problem of maintaining blindness. You don't have to unblind, but you do lose placebo. That group, the group that's been put into that blinded crossover system, you won't have a placebo group anymore to compare for purposes of.
That's right. You lose the contemporaneous placebo. That's right. You retain the value of having had a placebo at the beginning, though, because of the deferral, and that if there's a waning, particularly on the issue of waning immunity, obviously that difference might continue over time. So you retain some of the value of having a placebo at the beginning, but not all of the value, because things like particularly safety and other issues which might not be contingent just on the specific timing of when you got the vaccine, you will probably lose that or diminish that. You don't completely lose it. It depends on the characteristics of what it is you're measuring. There are many things we can look at still with this design, but that's why it has to be these designs coupled with the observational designs outside, which is obviously both what Pfizer has proposed, but also what the CDC and the FDA are also doing. So there's many things that can be learned outside the trial that we might have been looking to the trial to provide. Now we'll be depending more on observational designs if we move more to these trials. So we don't lose all the benefits of the placebo group, but we do lose some of them. but that may not be a practical option, which was my point.
Your position is that we should not move people into the blind crossover design until they're eligible in their particular societal group for getting it anyway. I will say that we have gotten a lot of comments from folks who are in trials, like dozens of them, and their line seems to be, well, if EUA is granted, I should be offered it. I'm not asking to opine on that, but my question is, your view is you should wait till it decidedly their group comes up, but if EUA didn't cover them at all, because that is one of the possibilities we might talk about later, would that help in the ethical determination that, yes, they should definitely wait, because after all, EUA has not been granted for their group yet anyway?
I haven't thought through that whole chain. All I will say is that they don't, again, I mentioned this previously, I don't think they are, it wasn't part of the initial contract. They understood that they could do without the vaccine for much longer, in fact, than they probably will have under a deferred policy. And they still get priority under this plan. It's just they get it first within their own priority group. So it's not that they are not owed anything. There is a reciprocity here in that when their, in a sense, turn comes up, they get it first through the trial mechanism. And so they're not completely, they're not disadvantaged by being in the trial. In fact, they're advantaged, just maybe not as much as they might want by demanding it today. But as I said, I don't think the ethical calculus is such, and other groups have not thought it such, that they are necessarily owed that just by virtue of being in the trial. But they do get a benefit.
Okay, thank you very much. I know these are very difficult situations and scenarios that we will need to be talking about again during the first part of our discussion. I apologize also to those people who I haven't been able to call on. You'll have your chance during the discussion because there's going to be a lot that we're going to have to talk about in the first part of our 2-hour period that we have assigned this afternoon. I am not sure, Prada, when should we come back? Because I know some of the public presenters are ready to go at noon Eastern.
Prabha,
Or
Kathleen. So we're scheduled to come back at, we're originally scheduled to come back around 12 o'clock, but we still have to call in all the public speakers. So let's just schedule it for right now, as long as Prabha, are you there?
Yes, I am here. And
okay, go ahead.
So, yeah, so we are scheduled to start at noon time. So are they all lined up now to go at noon, or do we have 5 minutes or extra to give them?
No, I need to call them in all now. So let's just, we will project to be scheduled for a 25 to 30 minute break, but we have to confirm all the OPH speakers come in. So let's, at this time, Prabha, if you agree, we'll do a 25-minute break, but we may have to extend it a little to make sure we get time to get everybody in. Okay?
Yes. Okay.
Okay. That's the guidance I needed. Thank you. So we're going to try to resume around noon Eastern.
All right.
Thank you.
So thank you. So with that, we are going to take a break.